Good day, everyone. I'm Max Hegblom, Editor-in-Chief of FEMS Microbiology Ecology, and it is my pleasure to welcome you to this webinar on biogeochemical cycling. We continue our webinar series, which enables us to highlight different topics of microbiology to a broad audience. Those of you who have tuned into our previous webinars know that FEMS, the Federation of European Microbiological Societies, invest in science, using the income from our journals to fund charitable activities and support our community. Learned societies and their journals provide grants to scientists, organize and support conferences, summer schools, and sponsor a range of events such as this webinar series. These provide a forum for the presentation and discussion of key research, enabling the flow of ideas to a worldwide audience. As an author, when you publish in FEMS journals, and as a reader, when you use these publications, you directly support the various activities in which FEMS invests back in science. If you missed our earlier webinars, they are available at the FEMS OUP websites. Indeed, I wish to thank the staff of FEMS and Oxford University Press for their work behind the scenes in making these webinars happen. Also, a quick reminder that the FEMS Congress is just around the corner. FEMS 2023 is the biggest event in Europe, dedicating to showcasing the latest development across a broad scope of microbiology. This takes place July 9 to 13 in Hamburg, Germany. During this Congress, you can learn from top-level scientists from 10 main Congress topics and connect with thousands of scientists. Register now and hope to see you soon in Hamburg. Today, we focus on the topic of biogeochemical cycles. As you know, microbes make the world go round. Whether it's carbon, nitrogen, sulfur, iron, or arsenic, or the many other elements, microbial processes are central to the movement and transformation of chemical elements and compounds. Indeed, there is much to learn of the interactions and biological, geological, and chemical processes involved in these cyclings. Our three speakers will dive into this topic, focusing on three different aspects of biogeochemical cycles in different habitats. First, Tilman Luders, Chair of Ecological Microbiology from University of Bayreuth, Germany, will focus on tracing long-distance electron transfer by cable bacteria in freshwater sediments. This will be followed by Martina Glodowska, Department of Microbiology at Radboud University, examining nitrate leaching and its implication for iron and arsenic mobility. And finally, Ahmed Pinto from the School of Civil and Environmental Engineering, Georgia Institute of Technology in Atlanta, USA, will talk to us about the role of Comamox bacteria with complex nitrifying communities. So again, welcome to our three speakers and welcome to all of you to this webinar. So first, Tilman.
Hi, can you hear me and see me?
Yes, works well.
Okay,
and,
so I will share my slides now.
The platform is yours.
Okay, you see the slides and also my mouse?
Yes, all good.
Looks good. Okay, thanks, Max, for inviting me. Hello, everyone around the globe. Thanks for joining our webinar today. I'm excited to present the first paper that is being presented here today. My name is Tilman Luders from University of Bayreuth, and I will be talking to you about some intriguing observations we made about tracing cable bacteria. I'm sure most of you have heard about cable bacteria, what they are, what they do in freshwater sediments. So the paper that I'm presenting here today was published last year by my former PhD student, Corinna Sachs. It's a great study that I think opened some interesting new doors into understanding of cable bacteria ecology in freshwater sediments. And it's my pleasure to present this paper here on her behalf today. Maybe she's watching, I don't know, but she's now in industry. So it's my pleasure to continue also on this research here in Bayreuth. So it was tracing long-distance electron transfer and cable bacteria by an agar pillar gradient column. This is something that I'll be introducing to you in a minute. But first, let me briefly introduce the discovery of cable bacteria. Most of you will know that they were discovered in the early 2010s. Here, this was this pioneering paper by Lars Peter Nielsen's group on the discovery of cable bacteria in Aarhus Bay harbor sediments. Cable bacteria are electrically active populations of filamentous bacteria. It's actually phylogenetically, it's sulfate reducers, Desulfobulbaceae, but they are actually operating in reverse. So they are oxidizing sulfide, respiring oxygen, and they are capable of doing this in a spatially decoupled manner. So they can conduct electrons that they harvest from the oxidation of sulfide at a depth in the sediment. They can conduct these electrons over centimeter distance to the cathodic side of the filament, where they can reduce oxygen. And this cathodic oxygen reduction causes a very characteristic pH peak. And that's also some of the early signatures that were first discovered there when the Danish colleagues started looking at these Aarhus sediments. And that's how they kind of figured it all out. So cable bacteria are known to be very abundant, widespread. Here you see some marine sediments, a dense network of filamentous conductive bacteria sitting here. They have they are gram-negative, Desulfobulbaceae. They have a joint periplasm where they have some conductive structures, conductive fibers that are then conducting these electrons from the anode to the cathode of the filament, and that's how they kind of beat other sulfide oxidizers for competitive for competitive hydrogen sulfide. Also important to mention that there is no pure culture yet, and there's also not too much work that has been done on cable bacteria in freshwater systems. So that's where we also started going. So that's what was basically one of the early motivations of this work that I will be presenting to you today. So, but there is some papers out. So the first paper, again, from our dear Danish colleagues, this was in 2015, where Lars Peter Nielsen, basically in the backyard of the place where he lives, he has a little stream, the Gibber, and also there they discovered a high abundance of cable bacteria. Here you see the length density up to 40 meters of filaments per cubic centimeter of cable bacteria in a little freshwater creek in Denmark. Before I moved to Bayreuth some years ago, we were working in Munich and also got going on this question of whether cable bacteria could also be found in groundwater systems, in aquifers, and especially also in polluted aquifers, where we suspected that they could have either an indirect or a direct role in the degradation of organic pollutants. So also here we incubated sediments, and then we saw this establishment of this very characteristic hypoxic zone of up to several centimeters within weeks and months. And when we did microsensor measurements, again, we saw the same pH peak here, a characteristic for the cable bacteria activity, and also a very pronounced hypoxic zone. And then when we looked at the microbes, then we saw that these Desulfobulbaceae were highly abundant in these laboratory sediment columns, right to this depth of highest oxygen respiration. So also in groundwater and also in polluted aquifer sediments, there seems to be a role for cable bacteria. But the work that I would now like to present to you today is trying to get a more wider angle, not just looking at groundwater, but also looking at some freshwater systems here in Bavaria. We would like to learn whether there is also whether also here we can find cable bacteria. What is their diversity? How are they distributed? Is there some characteristic distinctions between the cable bacteria that we see in surface water sediments and also in groundwater? Can we maybe enrich them with some tricks? Are they maybe directly or indirectly involved in pollutant degradation? And the long term, hopefully, maybe there can also be some potential for the application of these intriguing microbes for organic matter degradation or pollutant degradation in a modulated way. So, the first most important question or objective we had was, so how can we actually work with cable bacteria in freshwater sediments without killing too many microsensors? So because our sediments, our freshwater sediments, lake and also groundwater sediments, they are much more mineral. So it's much more difficult to work with these fragile microsensors compared to the highly organic marine sediments where they were first discovered. And that is the idea. That was the idea of Corinna and also Diraj, a postdoc working at that time in my group. So here we introduce the agar pillar column. So this is basically a sediment column where before we fill in these sediments where we hope to detect the cable bacteria, we cast a central agar pillar, which then equilibrates via diffusion with the surrounding sediment. We can have space for an amendment at the bottom. So here we can either add iron sulfide or also pollutants and all of this. And then we can then via the sediment kind of develops the hypoxic zone and the populations of the different microbes develop. We can then repetitively measure via the microsensors into this agar pillar. So that was the idea. And we set up microcosms with 3 different inoculars, so that one was from the Isar, or a little stream next to the Isar, heavily impacted by agriculture, then a highly sulfidic lake in the northern Alps, and then again this polluted aquifer sediment from Düsseldorf, where we had already been working previously. So that was the experiment. And here you see a lot of microsensor data. We don't have the time to really go through the details here today. On the left side, you see the patterns in the agar pillar. On the right side in the sediment, and both then right after the start of the column incubation and then after some weeks, so 28 days. And the most pronounced signatures of long-distance electron transfer were clearly apparent. So this is mostly a very pronounced hypoxic zone that was clearly detectable here in the sediment. Not so much in the agar pillar. So there's also a hypoxic zone, but it's not as pronounced. And that's also not unexpected, because we expect that both oxygen and also sulfide will diffuse more efficiently in the agar pillar compared to the sediment. But what was very encouraging was that over time, we saw a very strong electric potential. So that's also a microsensor measurement that was not there initially. And this measures the electric field that is generated via the long-distance electron transfer. So this electric potential was a clear indicator that we have electrically active microbes that had been growing in our sediment columns, and even more interestingly, also in the agar pillar itself. Okay, so we move on to the lake sediment. So that was the Alatsee, same picture here. We have some minor differences, but we also have a very strong hypoxic zone. Here even we do not see the sulfide in the agar, and we have an even stronger electric potential that was detectable here in the sediment columns from the Alatsee. So very encouraging. And the third set of microcosms was the polluted aquifer. And here things look very different. So we have no electric potential in the agar pillar from the aquifer sediment. we have a slight increase in electric potential in the sediment, but clearly not as strong. So maybe a factor of 5-fold less electric potential, electric field compared to the surface water sediments that I just showed to you. But also here we see oxygen penetration, we see a hypoxic zone, but there was clearly not as much sulfide coming up from below in the aquifer sediments. So these were the major distinctions that we observed between the surface water sediments and the groundwater sediments that we incubated in our agar pillar columns. And then next thing was that we did sequencing. So we sliced these columns after incubation and we did the sequencing. I will show you in a minute also that we didn't do Illumina sequencing, but bio long read sequencing, but that's not relevant in this graph. Here you see on the left, here first you see the original inoculum, and then you see the agar pillar as it was sliced in millimeter and centimeter intervals, and here also the sediment over depth. So you see colorful bar graphs. I just highlight the most important message. So what we see here, and that was very surprising, we see a high enrichment of these desulfobulbaceae reeds within the agar pillar itself. And next to that, co-localized, there's some Clostridia, so Ruminococcaceae. These are well-known fermenters, carbohydrate degraders. So somehow there seems to be some connection here. Maybe also these fermenters can plug into these electrically active filaments and also use them as an electron sink. And these cable bacteria sequences were up to 10-fold enriched highly significantly in the agar pillar compared to the sediment. So there's also desulfobulbaceae here in the sediment, but they are clearly much, much less abundant. Okay, we go on to the lake sediment. Intriguingly, or regrettably here, we lost some very important depth of the agar pillar, because this was when we started to slice it, it was somehow falling apart, it was disintegrating. So we don't have the complete picture here, but comparing the electric potentials that we see between the lake and the stream sediment, we expect that the picture would have looked similar. So we should also have seen this enrichment of cable bacteria, of the desulfobulbaceae there. What the difference was that we see also a higher abundance of the desulfobulbaceae in the sediments compared to the stream sediment. And also we see some other intriguing microbes sitting in the agar pillar, like these aeromonadaceae, which are also known, or at least some of them are known as aerobic sulfide oxidizers, but they were detected at a depth where there was clearly no more oxygen. So also this would have required, their activity would have required some plugging into electrically active filaments of cable bacteria. Okay, and the third set of columns, this was the aquifer sediments, and here the picture looked completely different. We had no cable bacteria, no desulfobulbaceae in the agar pillar itself, while we had a much higher abundance, and especially with depth, of these desulfobulbaceae within the sediment. They were co-localized with other well-known sulfide oxidizers, the thiovulaceae, sulfuricella. So there seems to be some sharing of labor going on here, maybe, or some interactions, competition in sulfide oxidation within the sediment, while the agar pillar looked very different. We had again fermenters, we had again aeromonadaceae, but also other guys sitting here on top in the first few millimeters, so we don't really understand what has been going on in this pillar. It was clearly not dominated by, or it didn't show this enrichment of cable of desulfobulbaceae, and also not of other sulfur oxidizing lineages. Okay, we could also image these filaments with microscopy, fluorescence, in situ hybridization, also just here in the agar pillar down here. So up here you see the sediment. This is DAPI, and with Desulfobacteriaceae, Desulfobulbaceae probe. Here you see this nice filament in the stream sediment, also in the lake sediment, and here just a DAPI image of the aquifer sediment, where we also have these long filaments growing nicely. Here in the polluted aquifer, then of course we are not successful looking into the agar pillar, but we did see the filaments also growing inside the agar pillar of the surface water sediments. So that was quite a surprise. And now the last part of this this experiment, showing you that with these PacBio full-length amplicon sequences that we generated, we can actually also do really proper phylogeny. So we received up to 14,000 high quality reads, these circular consensus sequences of almost full-length 16S amplicons after QC. In average, our reads had a read length of over 24,000 bases. That means 17 passes for each amplicon. So this is highly confident. This gives us high confidence in the data that we use then for calculating phylogenetic trees. And what you see here is phylogeny of the Desulfo bulbaceae and within the Desulfobacter rota. And up here you have these two proposed species lineages of genera and species of the known cable bacteria, that is Electrotrix, Candidatus electrotrix from marine sediments, and Candidatus electronema from freshwater sediments. And in here you see these clusters that we found within our incubations. And here we can show that we had at least 3 species level clusters that were below these proposed thresholds of species identity within the Candidatus electronema genus. And the groundwater cable bacteria that we found within the Düsseldorf sediments, they were, as already previously shown, very different, much more deeply branching, more similar to the Sulforibibrio and other sulfur cycling microbes here further down at the root. Okay, the last 2 slides I have for today is on a SIP experiment that we then added to this approach to ask the question whether these cable bacteria could also be directly or indirectly involved in pollutant degradation within these sediments. And Corinna set up a experiment where she did comparative labeling columns, where some of them had filters as a barrier for the growth of cable bacteria in the top sediments. And so we incubated sediments with or without these filters, and then added 12 or 13C toluene from below, and then looked at what would happen and who would be degrading the toluene. And the first result would be that with the filter, this reduces our emission of 13 CO2 into the headspace. So we see that with the filters, so with a prohibited activity, without activity of cable bacteria, we get less CO2 production from the toluene. So that could also mean that there is less biodegradation. And down here we see one depth profile. They were highly comparable between 12 and 13C microcosms. So I don't show all of this data, but we have a clear hypoxic zone and also a bit of electric potential that was developing here, not as pronounced as previously. But then we extracted the DNA, and that's a complex graph, but I just have like 2 messages or a very simple message. So here we have the sequencing libraries always from heavy to light. So the labeled taxa would always be sitting here in these heavy fractions. Here there's no labeling. Here we have 13C and here with filter. And what we saw in the labeled DNA was then unfortunately not very, so it was a bit of a disappointment. So we didn't see any role of Desulfobulbaceae. So they were there, but they were of low abundance, and the microbes that were highly labeled here in the heavy DNA, these were known aerobic toluene degraders within the Rhodocyclaceae or Moraxellaceae. But the Desulfobulbaceae in these columns, they unfortunately did not pick up any label. Still, there is still the option that there was an indirect stimulation of biodegradation when these cable bacteria can kind of overcome diffusion limitations within redox gradients or at interfaces. So that brings me to the conclusions. So the cable bacteria seem to be much more widespread in freshwater systems than we do know. We saw patterns of long-distance electron transfer in all the sediments that we investigated. The agar pillar seems to be a very intriguing selective niche for actually pulling out these these electrically active microbes. We found at least 3 new species-level taxa with our full-length amplicons. There was a very fundamental distinction between the groundwater cable bacteria. They did not grow into the agar pillar, and this is in line with distinct ecophysiologies that have already been proposed. And we didn't find any involvement in organic pollutant degradation. But there could be lots of interactions going on also in the agar pillar with other heterotrophs, fermenters, with other sulfide oxidizers. So that clearly shows that there's an untapped potential of cable bacteria in all kinds of redox processes and maybe also restoration of organically burdened sediments. And that's a perspective that we are currently trying to develop onwards in our ongoing research. So with this, I would like to thank, first of all, my former group in Munich some years ago here, where Corinna started her work, our colleagues at Aarhus University who supported us and collaborated very efficiently, and also the people that were involved here in the part of the research that then continued here in Bayreuth. With this, I'd like to thank you for your attention and happy to take some questions later on.
Thank you, Tilman. Again, the wonderful talk, and the cable bacteria are just so amazingly interesting. So fun to hear an update on this. Again, we will move all the questions till the very end. So please, I know that there were a number of hands up already. So if you type in your questions in the Q&A, we'll get back to these then at the end of the webinar. And so we will move on to Martina Glodowska. And again, another very interesting question on arsenic mobility, the interplay with iron, and issues, of course, in the Red River of Vietnam. So Martina.
Yes, hello everyone, and thanks Max for a nice introduction and for inviting me to give this talk and organizing this webinar. I think it's a really great initiative. So let me share my presentation. All right. Okay. Okay, do you see it?
Yes, all good.
All right, so yeah, as Max already mentioned, I'm going to tell you a little bit about how nitrate leaching can affect iron and arsenic mobility in the aquifer. But before we get into that, I would like to tell you a little bit and to tell you what is the source of arsenic and where does it originate. So generally, we can have arsenic originating from anthropogenic activity, but in most of the cases, actually, it comes from geogenic sources. So we can commonly find arsenic in the earth crust. It's actually 20th most abundant element in the earth crust. And so it's commonly found in the different minerals and different rocks. And well, in South and Southeast Asia, which is the region that is particularly affected by arsenic groundwater contamination, well, it often originates from Himalaya mountains, where eroded sediments and rocks are basically washed down the mountains and deposited in river deltas, leading to the formation of these aquifers. And normally these aquifers are relatively stable. The arsenic is immobilized in the solid phase. But because of the changes in biogeochemistry, eventually this arsenic can be mobilized to the groundwater. And of course, this is a big problem because well, South and Southeast Asia is a very densely populated area, and consequently, it can lead to the contamination of water and affect the livelihood of 220 million people that can be potentially exposed to the high concentration of arsenic. And I think I don't need to tell you that arsenic is a potent cancerogenic metalloid. It's a human one cancerogen. leading to the serious health issues, as you can see on the picture, usually expressed in skin lesions, eventually leading to the different type of cancers, more frequently skin cancer. So of course, that was one of the main motivation on studying this processes leading to the arsenic groundwater contamination. So generally, we have many different mechanisms on how this arsenic can be mobilized from sediment to the groundwater. This has been done already previously. Lots of study were tackling this problem. And so one of this mechanism can be due to the oxidation of arsenic-rich sulfides, iron sulfides such as pyrite, because these minerals, they have a very strong affinity to arsenic. And when they are oxidized, they can simply release arsenic to the groundwater. Another mechanism is via competitive sorption and exchange of arsenic with phosphorus. Because if you look at the periodic table, you would quickly realize that phosphorus and arsenic are very similar to each other. So they are kind of like analogs. And if we have an input of phosphorus, for example, to the aquifer, well, this phosphorus will compete with arsenic for absorption sites on the iron hydroxide minerals. And often it will lead to the washout and release of this arsenic to the groundwater. And then it can be also mobilized via changes in absorption capacity of iron oxyhydroxides, and for example, triggered by changes in the pH. But one of the main mechanisms that are believed to drive this massive release of arsenic is via microbially mediated reductive dissolution of arsenic-bearing iron minerals. And well, this is the process that I have been extensively studied during my PhD. So we tackle this problem from many different angles. And well, this process is relatively well understood already. So we know a lot about microorganisms that can mediate this process. These are usually heterotrophic iron-reducing microorganisms that can basically use different type of organic carbon, oxidize this carbon either to carbon dioxide or other intermediate carbon compounds. And well, they use iron 3 as a terminal electron acceptor. And so they drop the electrons on iron 3 minerals. And by doing this, they basically drive a reductive dissolution of these iron minerals. And by this, they also co-release arsenic that is sorbed to these iron minerals. So this process is relatively well understood. But well, although we answer many questions regarding how this process is happening, there are also many new questions that came along. For example, what will happen if there will be input of nitrate to the aquifer, for example, originating from agricultural activities? Because, well, nitrate is also quite attractive electron acceptor. It's actually thermodynamically more favorable than iron because it can provide a higher Gibbs-free energy to the microorganisms. So if the microorganism would preferentially go for a nitrate rather than iron, well, that would potentially inhibit the reduction of iron and also mobilization of arsenic. So that was also the reason why we conducted this research. We wanted to see what would actually happen in under more environmental condition and study. And so luckily, we've been working in Vietnam. Well, during my PhD, we performed several sampling campaigns in Vietnam in a small village of Van Phuoc, which was located about 15 kilometers south from Hanoi in this meander of Red River Delta. And well, this area has been already very extensively studied. So it was actually great because we had so much information about the geology, hydrology, and geochemistry of this aquifer. So, yeah, we preserved lots of sediments from these drilling campaigns. And so these sediments we could use for our experiments. So from all this previous study that we conducted, but also all other research teams that were working in this area, we knew that this Van Phuoc aquifer is quite peculiar because it's kind of divided between Holocene aquifer, much younger aquifer that was known to have a very strongly reducing groundwater, very highly enriched in arsenic and iron, and also very methanogenic. So we had a very high concentration of methane in this Holocene aquifer. On the other hand, we had also Pleistocene aquifer, which is characterized by groundwater that is rather suboxic. And this water is rather pristine because the concentration of arsenic, iron was very low, and the methane was nearly undetectable. And these two aquifers were divided with the redox transition zone. And this transition zone was believed to be this place where all this biogeochemical processes leading to a mobilization of arsenic were taking place. On top, the aquifers were actually covered with a silty clay aquitard. And from the drilling campaigns, we knew that this aquitard contained these lenses of organic matter. And from our previous work, we also knew, based on our study focusing on water isotopes and carbon isotopes, we knew that some of this water from the top, as well as the carbon embedded in this aquifer, could actually percolate and leach down to the aquifer. And so that was also the motivation for the study, because, well, on top of this aquifer, we had a lot of agriculture happening. So there is a lot of banana plantation, lots of animals, aquaculture. And of course, when there is agriculture, there is always a fertilizer that come with it. And so if there is application of fertilizers, well, most likely if the water can percolate and penetrate, well, probably also some of this nitrogen compounds, such as nitrate, can also leach down to the aquifer. So that was our question: what will happen to the arsenic and iron, and if we have an input of this additional electron acceptor? And because it's a methanogenic environment, well, what will also happen to methane, and whether we can see any nitrate-dependent anaerobic methane oxidation? So what we've done, well, we had the sediments that we collected from our previous sampling campaign. We drilled several cores all the way up to nearly 50 meters below ground. And we used the sediments from about 30 meter depth. So these were very orange sediments, sandy sediments, and very rich in iron oxyhydroxide, because these sediments are also known to retain lots of arsenic. As I mentioned, this iron oxyhydroxides, they have a high absorption affinity toward arsenic, so they are known to contain lots of arsenic. And so we use the sediment to run the different microcosm setups. So we have our control, where we simply use the sediments with the artificial groundwater. Then we also supplemented it with label nitrate and label methane. Then we also wanted to know what would be the effect of nitrate-dependent anaerobic methane oxidation, because if we have an input of nitrate, we would also expect that this endamo process will eventually take place. But to make sure that it will happen, we wanted to also add it eventually the inoculum from our enrichment cultures, because in our lab we have two of this endamo enrichment cultures, endamo from Oipolder, Dutch ditch. So we had this enrichment that consists mainly of Candidatus methanoperendens and Candidatus methanomirabilis. So this microorganism basically can oxidize methane to carbon dioxide and reduce nitrate to nitrite. And then methanomirabilis take up the nitrite and then further reduce it to nitrogen gas. Then the other enrichment cultures that we have in our lab originate from rice paddy soils from Italy. I call it Endamo V. It's mainly Candidatus methanoperendens, different than the one in Endamo O, because it can basically do all the way the nitrification from nitrate to nitrogen gas with the methane as electron donor. So I use this enrichment cultures and inoculate the sediments and to see what will be the effect of this endamo activity on iron and arsenic mobility. And we also have a biotic setup where we, besides adding the biomass, we also added sodium azide to really see whether biomass itself drive any biogeochemical processes. So yeah, let me show you some results of this experiment. So first we wanted to see what will happen to nitrate. And so what we observed is that actually in all of the setups, we had a very quick reduction and removal of nitrate. So in nearly less than 10 days, I think in 5 days, about all of the nitrate was almost completely depleted, except for a biotic setup with the sodium azide. And so that will tell us that our native microbial community, because also we see this removal in microcosms where we added nitrate and methane, but we did not inoculate it with endamo activity. Well, native microbial community seems to be very efficient in the removal of nitrate. And of course, as expected, endamo enrichment also stimulated a very quick removal of nitrate. However, when we look at the nitrite, we could see that native microbial community did not appear to be capable of further reduction of nitrite and further to other nitrogen compounds. We had actually quite a big spike of nitrite, and then, well, a little bit of it was reduced, but the majority remain in a bubble. So it means that native microbial community was simply incapable of reduction of nitrate, and while our endomoculture, of course, we knew that they are capable of doing so. When we look at ammonium, we have an accumulation of ammonium in our abiotic control, and this is due to the addition of sodium azide, but also the composition and breakdown of biomass added of the endomoculture. We also observed the increase of ammonium in our inoculated sediments with endomocultures, and that could be either also because some of this biomass eventually died out and was mineralized, but it could be also because we know that the Candidatus methanoperennens species and also are capable of performing the dissimilatory nitrate reduction to ammonium. So it is also possible that some of this nitrate was actually converted to ammonium. And then when we look at the label nitrogen gas, we could see that it's mainly our endomoculture supplemented sediments were mainly converting the nitrate all the way to nitrogen gas, and this is what we expected. Well, the native microbial community clearly converted nitrate mainly to the nitrite, but it seems like that some fraction of it was also converted to the nitrogen gas. And then when we look at the methane concentration, it was pretty clear that it's only enrichment cultures that we added were performing a methane oxidation. We could see that there was a decrease of methane, and it was pretty much positively correlated with a nitrate concentration, while a native microbial community did not appear to really consume any of this methane. And it was also confirmed with 13 CO2 data, where we could see that there was an increase of labeled CO2 in our enriched sediments, endamo-enriched sediments, while it was pretty much stable in all other setups. And so with that, we could clearly see that methane oxidation could be only performed by endamo-inoculated sediments. However, the native microbial community did not really show the potential for nitrate-dependent anaerobic methane oxidation. And when we look at the iron concentrations, that was something that was particularly interesting for me, because what we could see in this result is that when we added nitrate to our sediments, clearly this inhibited the iron reduction, because we did not really see any mobilization of iron to the aqueous phase. There was no also an increase of sedimentary iron too in the sediments. So that was clearly the effect of additional electron acceptor that was competing with the iron, and most likely was preferentially used by our microbial community, native microbial community. Surprisingly, we have a lots of iron being reduced and mobilized to the water, but also increase in a sediment and in the presence of our endomocultures. And so that would clearly show that our endamo enrichment cultures are also capable of iron reduction. And this is not surprise, because we know that these Candidatus methanoperennens archaea are capable of coupling iron reduction with methane oxidation. We also had quite a bit of iron being reduced, particularly in the sediments. Well, in the sediment, we had an accumulation of iron too in the sediments and our control, meaning that there are native microbial communities that could also reduce iron with whatever organic carbon that was still present in the sediment. Sadly, we also have a little bit of iron reduction in our abiotic control, and that would indicate that probably we fail to completely inhibit microbial activity with a sodium azide, and it was mainly the endamo iron reducing capacity. And finally, we also look at the arsenic mobilization, and that was also rather surprising results, because as we saw, actually iron reduction was the most pronounced in endamo V enriched sediments. But surprisingly, there was no arsenic being mobilized in the presence of this this enrichment culture. And however, it's the endamo O that seems like stimulated the most arsenic mobilization. And what was also something that I was very pleased to see is that, again, with the nitrate, as we also saw, there was no iron reduction, there was also no arsenic mobilization. So again, with the input of nitrate, it seems like it inhibits the iron reduction and consequently arsenic mobilization. We also look at the manganese because we knew that manganese can be also potentially used as electron acceptor by this methanoperendin species. And what was also interesting to see is that this endamo V enrichment culture appeared to be able to use manganese as electron acceptor. Whether it was coupled with a methane oxidation, we are not entirely sure, but clearly a substantial amount of manganese was actually reduced in the presence of this culture. So yeah, endamo can stimulate the arsenic mobilization, at least this endamo O enrichment culture. Arsenic mobilization is inhibited by nitrate, and our culture seems to be also involved in a manganese reduction. Finally, we also look at the microbial community. We did the 16S amplicon sequencing to look at the archaea and bacteria. So what was particularly interesting is that our in situ sediments at the beginning of the experiments, they were already quite enriched in Candidatus methanoperendin species. And this is something that we already knew, that and these microorganisms are present in the sediments because we showed that in our previous study. But clearly this methanoperendin species were very different from the one that we introduced with our inoculants from enrichment cultures, because the native methanoperendin species were just not able of nitrate-dependent anaerobic methane oxidation. And then when we look at the bacterial community, well, in the endamo O enriched sediments, we had an increase of methylomirabilis species. So this is the nitrite-dependent methane oxidizer. And so this was not a surprise because it was introduced with the inoculum that we added, as it contained nearly 30% of it. With endamo V, we had a various the nitrifying community that got enriched, also Geobacter, that was putatively also involved in this iron reduction processes with the nitrate and methane supplemented microcosms. We have lots of microorganisms involved in nitrogen cycling, and in a control Geobacter. And this is something very consistent that we always see when we incubate these sediments. We always see a Geobacter being enriched when we don't add any type of organic carbon or other electron donors, and Geobacter will always pop up and will be always very abundant. So yeah, so with that, what we could conclude from this study is that our native microbial community is certainly capable of nitrate reduction. And well, this what it tells us is that if we indeed have a percolation of nitrate into the aquifer and input of this nitrogen from fertilizers, for example, well, good news is that it will inhibit the iron reduction and prevent arsenic mobilization. But the bad news is that, well, this community is not capable of denitrification. And so we end up with accumulation of nitrite, which is, of course, not a great thing in a groundwater either. And well, our endamo cultures also seems to be having effect on iron reduction. They both seems to be capable of iron reduction, and they seems to be also capable of mobilizing arsenic, at least the endamo O culture, while endamo V seems to be involved in a manganese reduction. And so with that, I would like to particularly thank my master student, Ying Xiao, that was behind this experiment. And he was actually the one that conducted all this incubation, but also all my previous team with whom I was working with in Vietnam, and we did lots of work together. And my current research group at Radboud University, and of course, my funding agency via SIAM. And yeah, I will be happy to take your question also in the end of the session.
Okay, thank you, Martina. I will do my best to keep my questions until later as well. I have quite a number for you, so I will suppress.
Yeah, I know
my urge
you've been
to-
working on arsenic, right? So
Yeah,
I
so I will
expect some
suppress
questions
my
from
urge
you.
to ask you now, but thank you. But again, please add your questions to the Q&A and we'll come back to them at the end. So with that, it's my pleasure to then introduce our final speaker for this webinar, Amit Pintot from Georgia Institute of Technology, who's continuing our topic on nitrogen and looking at the activity of Comomox bacteria. So Amit, the floor is yours.
Can you see my slide share?
Yes, they're fine.
Okay, great. Thank you. Thank you so much for the invitation to present here. And I'm glad to have this opportunity to talk to everybody about the work that we've been doing over the last few years related to complete ammonia oxidizing bacteria. As Max suggested, introduced me, my name is Amit Pinto. I am at the School of Civil and Environmental Engineering at Georgia Institute of Technology in Atlanta. So just a quick introduction to provide some context of where we are coming from when we look at the nitrogen cycle. My research group at Georgia Tech, we primarily are interested in microorganisms in the engineered water cycle, whether that's in drinking water treatment, whether that's in wastewater treatment, whether that's during distribution of drinking water to homes from the drinking water treatment plant. That's our focus within this loop. That is the engineered water cycle. And our goal for the most part is to develop management frameworks to maximize the benefit and minimize the detriment of microbial communities. So as you can imagine, depending on where you are in the engineered water cycle, the impact that microorganisms can have on the quality of water that is being consumed or the quality of water that is being discharged into the environment, it can matter a lot. We work on several different topics on within the engineered water cycle at the intersection of microbial ecology, molecular biology, and design and engineering. But for today, I'm going to focus primarily on nitrification. Specifically, I'm going to focus on a paper that was led by my doctoral student, Dr. Catherine Velarde, who has just graduated, and she was supported by Dr. Irmari Koto. And this study was published last year in FEMS Microbiology Ecology, which is Comomox bacteria outnumber other canonical nitrifiers. And so to provide a little bit of context for those of us who are probably a little bit new to Comomox bacteria, some introduction to what these bacteria are about. Now, these complete ammonia oxidizing bacteria are relatively newly discovered when we think about nitrification and nitrogen microbiology historically. And what do I mean by relatively newly discovered? For a really long period of time, these are the nitrogen biotransformation pathways. And I'm not talking about organic nitrogen here, but inorganic nitrogen biotransformation pathways that we, as engineers, designed our systems around for whatever endpoint that we were interested in. And these were catalyzed by ammonia oxidizing bacteria, nitrite oxidizing bacteria, denitrifying heterotrophs, anamox bacteria, and then in 2015, complete ammonia oxidizing bacteria were discovered. And so one of the things that we've been doing as engineers within the engineered water cycle is trying to figure out what is the role, what is the importance of complete ammonia oxidizing bacteria within the engineered water cycle. So a little bit of context to complete ammonia oxidizing bacteria. As the name suggests, they are capable of completely oxidizing ammonia to nitrate. They have the full complement of genes for nitrification. They have the AMO genes encoding for the AMO enzyme, hydroxylamine dehydrogenase, nitrite oxidoreductase. So they behave like a combination of ammonia oxidizers and nitrite oxidizers. What's also very interesting is that they're actually phylogenetic siblings of sort of pure nitrite oxidizers. Here you have complete ammonia oxidizing bacteria in red, nitrite oxidizing bacteria in blue, and ammonia oxidizing bacteria in green. And what you can see over here on this phylogenetic tree is Comomox bacteria are sort of distributed among strict nitrite oxidizing bacteria within the genus Nitrospira. So if you take a gene such as a 16S ribosomal RNA gene, at least so far what we know is it's very difficult to tell complete ammonia oxidizing bacteria within the genus Nitrospira. from canonical sort of nitrospira nitrate oxidizing bacteria. So you really have to look at genes that encode for the nitrification function, because these genes have unique phylogenetic signal, and so you can kind of separate them from you can separate Comomox bacteria from canonical ammonia oxidizing bacteria. These Comomox bacteria are also quite found in very diverse environments. Here is just an example of different ecosystems: drinking water treatment plants, freshwater, groundwater, soil, wastewater treatment plants. And this is a study by Alex Palomo from Bart Smeth's group, where they looked at the richness of Comomox nitrospira or NOB nitrospira, and they looked at the normalized abundance of these organisms. And what you can see is that Comomox nitrospira tend to co-occur with canonical nitrate oxidizing bacteria in a whole host of different environments, and also along with other ammonia oxidizing bacteria. The catch so far is that, as far as I know, we haven't yet seen them in marine environments. The other thing that we know, and that becomes quite important from an engineering perspective, is that Comomox bacteria tend to grow quite slowly. Here is an example on the x-axis of different wastewater treatment plants in this case. And on the top panel, what you're seeing is the retention time in these particular wastewater treatment plants. And on the bottom panel, what you're seeing is complete ammonia oxidizers as a proportion of total ammonia oxidizers. And as you see over here is as the retention time in the system increases, the complete ammonia oxidizers as a proportion of total ammonia oxidizers also goes up. So these are slow growers, and they like systems, they like biofilm-based growth, they like systems that have a long retention time. What's quite peculiar also about complete ammonia oxidizing bacteria is, even though they oxidize ammonia and nitrite, their affinity for ammonia and nitrite as compared to other nitrifiers is kind of different. And here there is only data from one isolate that exists in Michi Wagner and Holger Dames' lab in Vienna. And here you have the apparent affinity to ammonia, apparent affinity to nitrite, and this is Candidatus nitrospira inopinata. As you can see, it has very high affinity to ammonia and a very low affinity to nitrite compared to ammonia oxidizers and compared to nitrate oxidizers. And this becomes quite important in terms of the role that complete ammonia oxidizing bacteria may play in complex nitrifying communities. The last bit of background that I'm going to give about complete ammonia oxidizing bacteria is that they also have relatively diverse sort of metabolic potential. We've known for a long time that Nitrospira NOB, or nitrate oxidizing bacteria within the genus Nitrospira, can do a whole bunch of different things. And we use this for a long time to justify the fact that why are nitrite oxidizing Nitrospira bacteria so much more abundant in compared to ammonia oxidizing bacteria. And we attributed it to their ability to degrade urease, to supply ammonia oxidizing bacteria with ammonia, with their ability to use hydrogen or formate. And complete ammonia oxidizing bacteria also show some of these same traits in terms of how metabolically diverse and versatile they are. So what role do complete ammonia oxidizing bacteria play in complex nitrifying communities? And this schematic is a little bit complicated, so please don't pay attention to this. What I want you to, the big take home message here is Comomox are just one of the nitrifiers that are of engineering relevance, between ammonia oxidizing bacteria, anamox bacteria, nitrate oxidizing bacteria, and ammonia oxidizing archaea. So depending on where you are in the engineered water cycle, these bacteria may play very, very different roles, right? In wastewater treatment plants, we are interested in what is their role in ammonia removal. In drinking water treatment plants, we're interested in their role in ammonia removal in the drinking water treatment plant, but in the distribution system, we are interested in their role in how might they affect nitrite accumulation in drinking water. So their role and relevance really depends on where these organisms co-occur. So for today's talk, and tracing back to Catherine Velarde's paper, I'm going to talk about complete ammonia oxidizing bacteria in a drinking water biofilter. Now, this is a biofilter in city of Ann Arbor, Michigan, and you can see these biofiltration systems. It uses a combination of surface water and groundwater. And back in 2015, we had found complete ammonia oxidizing bacteria within clade A in these drinking water biofilters. And we've been studying these biofilters for a few years now. And so through shotgun metagenomics, what we had seen for this particular biofilter is it had a combination of aerobic ammonia, aerobic nitrifiers that tended to coexist together. So we've done, so as part of the study with the shotgun metagenomics, on these activated carbon media filters. And what we saw from this data was that there were 3 metagenome assembled genomes from Nitrosomonas cluster 6A. There were 4 genomes from Comomox bacteria, and these were in clade A1 as well as clade A2. So A1 was 1 mag and clade A2 was 3 mags. The A1 mag was very, very abundant relative to the other Comomox bacteria. And then finally, there were 2 lineage 2 Nitrospira nitrite oxidizing bacteria that were also present in there. So these were co-occurring. We wanted to see, what are the potential interactions between these aerobic nitrifiers. And for that, we went down into their genomes to try to assess what is their metabolic potential within these biofilters. And what we see is that there are ammonia oxidation genes clearly over here. Blue is Comomox bacteria over here. Orange is Nitrospira NOB, and red is. We see in this particular case is that genes associated with ammonia oxidation are present in Comomox bacteria. They're also present in ammonia oxidizing bacteria, but some of these genes didn't really bind together in the mags that we had, but we did find them in the metagenomes, and the phylogeny of those AMOA genes was consistent with the phylogeny of these mags. We were also able to see capacity to use urea or degrade urea in all the mags that we assembled. In Comomox bacteria, in nitrite oxidizing bacteria, in ammonia oxidizing bacteria. Now, the nitrite oxidizing bacteria, not all mags, genes associated with urea degradation were binned in them. But when we went back to the metagenomes, we could find these contexts that had genes associated with urea degradation that were classified as Nitrospira. So we presume that these Nitrospira had the capacity to degrade urea, but just that the genes were not assembled. So now that we have our setup in terms of what are these different nitrifiers that coexist together, we were very interested in these 3 questions: is what are the potential interactions between these aerobic nitrifiers? What role does nitrogen form play in mediating these interactions? So for example, if we provide them with ammonia, or we provide them with urea, all of them can degrade urea. What are we going to see in terms of the population dynamics of these AOB, Comomox, and NOB? And finally, what role does nitrogen availability play in mediating these interactions? That means what happens if you provide nitrogen in multiple forms at high concentrations, medium concentrations, or low concentrations? So the experiments that Katie set up was she set up these microcosm experiments where the activated carbon from these biofilters were incubated with media that they would experience in the drinking water treatment plant, amended with either ammonia or urea. And they were amended with ammonia at 3 concentrations: 14 milligrams per liter, 3.5 milligrams per liter, 1.5 milligram per liter, and urea such that the total nitrogen amended was 14, 3.5, and 1.5. And every week she would harvest biological replicates, and she would measure the nitrite concentrations, the ammonia concentrations, nitrate concentrations, pH, and all the other parameters that you can measure from this system. But then she would also harvest biomass to try to assess what is the abundance of these different nitrifiers in there. And for this, we took a look at the abundance of ammonia oxidizers. We took a look at the abundance of nitrite oxidizers and complete ammonia oxidizing bacteria as well. And if you take a look at the paper, there are some modifications that had to be done to the primers used to detect complete ammonia oxidizing bacteria, but those details are in the paper. So let's take a look at what happens. And this is data over a few months now. And just to orient you to the panels that we're going to see, on the y-axis and the x-axis, you will typically see the abundance of a specific nitrifying group. So over here, there is strict AOB relative abundance, and this relative abundance is estimated by dividing the qPCR total abundance of AOB by the qPCR total abundance of bacteria. So it is 16S in this case of ammonia oxidizing bacteria, qPCR data divided by 16S targeting total bacteria. And over here, you have nitrospira NOB, relative abundance. And these panels will tell you what the different conditions are that these were incubated in. So the first insight that we had is that the abundance and the colors will refer to the different treatments as well. The first insight that we had is that the abundance of strict Nitrosomonas like AOB and nitrospira like NOB were only strongly correlated in the urea-based microcosms. None of these correlations were very significant in the ammonia-amended columns. And one of the potential reasons this could be possible is that in addition to these uralytic positive Nitrosomonas, the nitrospira could also play a role in providing ammonia to these ammonia oxidizers via degradation of urea. So these nitrospira NOB also had genes associated with urea degradation. So it's possible that we are seeing stronger association between AOB and NOB in these urea amended columns because these nitrospira might be cross-feeding the ammonia oxidizing bacteria. But when we take a look at complete ammonia oxidizing bacteria and strict AOB, we did not really see any associations. And this was a little bit surprising. And I'll come to the reason for this a little bit later on. When we take a look at associations between strict complete ammonia oxidizing bacteria and nitrite oxidizing bacteria, what was quite interesting was that we never saw any significant association in the urea amended columns. Neither did we see a significant association in the high ammonia columns, microcosms. We did see significant associations in the microcosms where there was sort of low ammonia availability, low to medium ammonia availability, 1.5 milligrams per liter, 3.5 milligrams per liter ammonia, and nothing in the urea amended systems. So to give sort of a high level summary of these few months long experiments is, one, is there was no observable association between complete ammonia oxidizing bacteria and AOB. This was a little bit surprising. We thought maybe we would see some what would be suggestive of competitive interactions at those low ammonia, low urea systems. But there was potential what looked like negative interactions between complete ammonia oxidizing bacteria and NOB. These interactions or associations between NOB and Comomox bacteria were present when ammonia is supplied, but not when urea is supplied. And finally, these interactions were stronger and more significant when ammonia was at low to medium level, right? So we have to step back and ask ourselves this question. In this particular cascade of nitrogen biotransformations, why are there no negative interactions between Comomox and AOB? And why are there no, and why are the negative interactions or associations between Comomox and NOB only when the ammonia concentration is low? Actually, the correct question after some thinking is, why is there lack of negative association between Comomox bacteria and nitrospira NOB at high ammonia concentrations? And one of the things that we hypothesize is that when ammonia concentrations are high, because Comomox bacteria have this differential affinity for ammonia and nitrite, and their rates of ammonia and nitrite oxidation are also different, Comomox bacteria might actually be supplying nitrite to these nitrite oxidizing bacteria at high concentrations. These nitrite oxidizing bacteria also get nitrite from AOB. And so maybe we don't see this positive or negative association, but at low concentrations, when this supply of nitrite from Comomox to NOB drop, we start seeing these negative associations. So these negative associations actually might emerge from an elimination of potential sort of positive interactions between Comomox and NOB. Why do we not see during urea amendment? And over here, the possibility is that since all three organisms are capable of taking urea to ammonia, they might sort of independently be accessing that ammonia or nitrite from ammonia by regulating the rate of urea degradation. In fact, what we do see is that for some reason or the other, even though Comomox and all three are able to sort of degrade urea, Comomox bacteria really love urea-fed systems. Here you can see their abundance at the end, and this is in a paper that's in progress right now. At the end of the experiment, in the urea-fed systems, they really increase in abundance compared to the ammonia-fed systems, significant increase. And this is also corroborated by additional literature. We've recently also concluded a set of experiments. All the data that I've presented so far has been from batch experiments. We've also completed a set of continuous flow column experiments with the same setup, where the columns were subjected to 3 different ammonia, 3 different urea influent concentrations, and operated for a period of about 6 months or so. And we see the same thing, is that only in ammonia concentrations, ammonia-fed systems, do we see a negative association between Comomox bacteria and nitrate oxidizing bacteria, but we don't see any association between them in urea-fed systems. So if you would like to get a lot more detail about this work, you're welcome to take a look at this paper by Katie Velarde and Irmari Koto. You can also take a look at some of our other work that looks at interaction of complete ammonia oxidizing bacteria in drinking water and wastewater systems, including anaerobic ammonia oxidizers. And you can access those papers using this QR code. With that, I would like to thank you for your attention, for the organizers for inviting me to present over here, and for the previous speakers for very enlightening talks. Thank you so much.
Thank you so much, Amit. This is, again, very interesting. And again, thank you, Tillman, and then Martina as well. This is a really, really fun session, and we'll open it up for discussion and questions as well. I know, Tillman, you've already been answering some of them in the chat room, but there are, I think, a couple of things to come back to, I think, on the interactions with the cable bacteria. But just quickly, while I look and wait for others, also, I noticed that Lucian, Chow, and Gabriella, you have your hands up in the Q&A, maybe for a question, but as far as I know, we don't have a way to unmute you, so please type in your questions in the Q&A and we can come to that. But let me actually, going quickly to one, to Amit, just on the urea. So is this actually maybe sort of like a slow release ammonia source? And while providing urea, you actually never have high concentrations of ammonia. And with the high affinity of the Comomox, that would be the reason why they outcompete?
Yeah, I think that's what we are leaning to. We actually, when we run these, we've finished analyzing data from these columns, is what we see is when urea concentrations are very high, Comomox bacteria really get extremely enriched. And as the urea starts getting degraded, as you move down the column and ammonia is being formed, their concentrations start going down. So it's by regulating the production of ammonia, you can really sort of control how you enrich for complete ammonia oxidizing bacteria. I think that's precisely it. Yeah.
Okay. Yeah.
Yeah. And this has some engineering implications as well. So typically, when we think of a wastewater treatment plant, we think about most of the urea has been converted to ammonia as it moves from homes into the wastewater treatment plant. But as we start bringing those wastewater treatment plant closer to homes through decentralized wastewater treatment plant, for example, then you're going to have wastewater streams that are much more higher in urea than ammonia. And so the types of organisms that we design around as we move those wastewater treatment plants closer to the consumer, I think it's going to have some implications just based on the rate of ammonia formation.
Okay. And yeah, question to Martina, the nitrate-dependent methane oxidizers. So you didn't see that activity in the indigenous community, but of course, then adding the enrichment, that was active within these microcosms. Do you think it was just they are so difficult to enrich and the time frame in those experiments was enough, or are they just not there?
Yeah, it's true that these are very tedious microorganisms to get them in culture. It's also quite time consuming because these are slow growing archaea. So indeed, this is also possible that the time frame, because the experiment lasted only 60 few days, so perhaps that wasn't enough. We know that this, we had Candidatus methanoperennus species within the sediments, and we know from our previous experiments also that, well, we couple it to iron reduction, but that also took quite a long time. So it was only after 100 days that we started to see the methane oxidation, formation of labeled CO2 and iron reduction. So it is also possible that it would just need a much longer incubation time before we would actually start to see the nitrate reduction coupled to methane oxidation. However, yeah, you could see that nitrate was consumed so quickly, so we would need to continuously supply nitrate in order to keep that, or move to a bioreactor system.
So what about methane oxidation coupled to arsenate reduction?
Yeah, that's a hot topic, obviously. Well, as far as I know, there are some papers in a line probably coming out soon in a high impact journals. Yeah, so far it wasn't proof, but I know that from the last conference in ISME, I think Gene Tyson in Australia, they managed to couple it. So yeah, I believe that this is also a case. Well, in our enrichment cultures, we couldn't couple that yet. We are still working on that. Because we know that these metanoperandin species encode for arsenate reductases, and they have many copies of these genes. So I truly believe that they can do that. But I guess it's also quite slow process.
Yeah. As many of the very interesting processes are, so.
Indeed.
Patience. So there's a question for Tillman here in the chat, and this is regarding the features of the agar pillar column to compare to the regular sediment column. And again, what can we capture with this setup? I guess one is that you're able to actually analyze them, but does it actually change other aspects of that sediment profile?
Happy to comment on that. Yeah. So when we designed the agar pillar, our original idea was to simply have a diffusion equilibrated measurement highway for the microsensors without killing too many of them, because microsensors cost easily between 500 and 800 euros. So it's an expensive experiment if you ruin a number measuring into these columns again and again. But then, so the agar features are nothing fancy at all. It's just a 1.5% agar pillar that we cast inside a straw before we fill then the column with the rest of the sediment. And then we carefully pull out the straw like a cocktail straw, leaving the agar pillar behind. And then this can equilibrate sometime. There's nothing else. There's no nutrients, no medium or whatever. And then this agar pillar equilibrates via diffusion with the surrounding sediment. And we didn't expect that it would be efficiently colonized by microbes. So that was not our aim. But then when we saw that after the incubation of 4 weeks, the pillar was densely colonized with all kinds of microbes, including the cable bacteria, this came as a surprise. Yeah, but now, of course, we have to think how this diffusion space kind of acts as a selective niche, and also the matrix of the agar acts as a selective niche or habitat for the cable bacteria. And maybe we can make use of that to further enrich them, and and maybe one day culture them. We don't know.
Yeah, indeed. You mentioned in one of the online answers in the chat that they have not yet been cultivated, at least not in pure culture, but this suggests that this could be a way of actually selectively pulling them out from the rest of the sediment. So that's really interesting.
Yeah. So when these, could I ask a follow-up question? When these agar pillars are created, so are they amended with any nutrients? So
No.
maybe extracted nutrients from the soil media and
No,
create
it's just agar, 1.5%, and it just equilibrates with the surrounding sediment column.
Okay.
Yeah.
Very interesting. Cool.
So, but of course, the diffusion is more effective in the column compared to the much more tortuous sediment, and so we have a more efficient supply of oxygen and sulfide, so oxygen from above and sulfide from below, to whoever is living inside the column, and this is inside the pillar, and this is maybe what could have also contributed to this selection.
Yep.
Yeah, so Andreas Gombert's follow-up is whether the agar pillar works as a reservoir for anaerobic microbes.
as a reservoir for anaerobic microbes. So the whole column is anaerobic. So it's anaerobic below whatever 1.5 or 2 centimeters. So we believe that the pillar is a bit of a conductive highway to the oxygen on top. So maybe there's a benefit of living inside and having a more efficient interaction with the filaments, or the filaments themselves, the cable bacteria. And it is known that all kinds of aerobes can interact with cable bacteria, or microaerophils can interact with cable bacteria and use them as an electron sink. And this has also been proposed for fermenting microbes. So there's basically the idea that of these get in contact with these conductive fibers within the these structures within the joint periplasm of the filament, then maybe you can use it as for respiration. Yeah, and there's some nice papers out from the Danish colleagues. The most recent one just recently published by Kasper Kjeldsen, showing that the cable bacteria, they attract flocks of swarming bacteria that kind of seek contact with the filament and then swim off again, and that this gets shut off as soon as you destroy the electric connectivity.
So the others are using the cable bacteria as a way of getting rid of electrons then. Yep.
To plug in, yeah.
Yep.
Amazing, amazing.
So there's a question here from Olga Bernadette to Amit on other sources of nitrogen in especially then in wastewater, and that would be organic nitrogen from proteins. So how is that going to interact then with the Comamox and their interactions with other nitrifiers?
Yeah, that's a great question. I guess it's one, it would be relatively concentration dependent, is what where this nitrogen is, not just where it is sparked in terms of what compound it is, but what the concentration is. We have not looked into protein metabolisms yet. For example, the ability to degrade different amino acids or organic matter for complete ammonia oxidizing bacteria. So far, our focus has been the most sort of readily bioavailable forms of nitrogen that they might have. But this is a nice suggestion and something definitely to look into. Yeah.
Oh, check is there. I don't see anything else then that's unanswered, at least in the chat. So so still an opportunity. Anybody else here who has a final question? So Martina, a question back on the Red River, the Mekong, and other, and of course the key question there is really the mobility of arsenic as the concern. The rice paddies, and now recognize that rice is one of the main exposure routes to arsenic in the population. So how do you think all of these interactions of what's going on with iron reduction, nitrate reduction, arsenic mobilization, how can we use this to manage the rice paddies to reduce arsenic translocation?
Yeah, rice paddies are a quite different system compared to the aquifers, because of course they are much more organic rich. There are redox oxidation and reduction processes that are more dynamic in this systems, because we have a wetting season and we have a dry season. So it's very complex system. Yeah, I'm not really sure that we can really control it, because yeah, we understand this process better and better. But there are just so many different factors, because of course there are microorganisms that can oxidize arsenate, reduce arsenite, iron hydroxide on top of it. So all the reducing processes. So it's really so many different factors. Most of these processes are driven by carbon. So obviously, if we could control carbon inputs, probably that would be our way to get a little bit of better control on the arsenic mobility also. But there is no really way to get rid of carbon from the rice paddy soils. So yeah, it's quite challenging topic. So I'm not sure whether we have really any means right now to get rid of it. I mean, like obviously using different type of minerals that are having the strong affinity to our arsenic, such as pyrite, iron. Of course, we can use that, but they are just never stable enough for a longer period to really retain this arsenic for prolonged time.
Yeah, and of course, understanding where arsenic mobilization is really a major risk, and then thinking of other mitigations as well. So
yes, indeed. Well, I mean, it's really the well, in the case of groundwater, at least, it's the treatment of this water. So there we have a means of treatment. But in the rice paddy soils, yeah, this is much more difficult, because then we have also plants that uptake all of this. So yeah, this is much more difficult.
indeed. Any final questions, Tilman, Amit, Martina, from you? I think then we will wrap this up. Thank you so much for taking part in this webinar. This was really interesting. And again, thanks for your papers in FEMS Microbiology Ecology. And I want to thank everybody here who's been in attendance. It's been a wonderful audience from actually really around the world that I looked at on the attendance list as well. And again, thanks to the staff at FEMS and OUP for doing a lot of work behind the scenes to make all of this happen. So thank you, Joe, there in the back. And again, we will hopefully see many of you at the FEMS Congress or other in-person conferences in the summer, and stay tuned for the next webinar on another exciting topic in microbial ecology. I'm happy to take any suggestions as well. We haven't yet thought of where we're going next, likely in the fall. So until then, take care.
Thanks for having us, Max. See you in Hamburg.
Yes.
Thank you.
Bye.